What I'm going to go over now is just some names of organisms that are placed into certain groups based on their preferences for the environmental conditions in which they grow. So essentially, we have growth preferences. And some of these things are not just where the organism likes that environment, but is absolutely required to have a certain specific environmental condition, or else it will not be able to grow. So, for example, temperature. So here you have a degree centigrade temperature and a rate of growth. All right, and these curves represent different organisms that have a certain preference for a temperature range. Right, so organisms that tend to grow at very low temperatures we call psychrophiles. So psychrophiles like temperatures that are sort of between, say, 0 to 20 degrees centigrade. But the thing is, you can see, in general, for this particular organism here, it won't survive. So at, say, 30 degrees, it won't grow at all, right? So it absolutely requires very cold temperatures. And some can actually grow at even colder temperatures that allow them to get even below 0. Next, we kind of have a group of organisms that grow in this particular range here. This particular range being about like 20 to 45 degrees centigrade, and these are our mesophiles. I'll highlight that one because the organisms that are part of our microbiome are all considered mesophiles because they would live in our bodies or on our bodies, so they have to be living at these temperatures, and they would typically require those temperatures. If they get much warmer, they wouldn't survive, and if they're much colder, then they won't survive or be able to grow. Then we have organisms that start to prefer much warmer temperatures, temperatures that potentially could cause proteins and some of these others to denature. These are the thermophiles. These organisms are like 55, let's say, to 85 degrees centigrade. And then in a minute, I'm going to talk about some of the adaptations that some of these organisms will need, because now we're talking about conditions where proteins generally would denature at some of these temperatures. And so there has to be some biochemical differences within these organisms, not just to operate, but even just to survive and maintain the integrity of the cellular structure, of their enzymes and everything else at these temperatures. And then you have some that are really up there. We have your hyperthermophiles. These organisms can survive at temperatures in excess of 100 degrees centigrade. So this is like around 85 to 113 degrees centigrade is like this stated range. Again, a lot of additional modifications to the cell membrane in order to stay intact are going to be required again and again to the proteins and enzymes. So, excuse me, I'll give you a sort of example. I'll talk about the heat ones first. At high temperatures, there's a whole variety of different biochemical adaptations that have to take place, and I'm really only going to talk about just two as just an example of them. One will have to do with the cell membrane. So at high temperatures, we're going to tend to see a lot more of the saturated fatty acids as the tails for the phospholipids. And if you don't recall much about the structure of phospholipids and membranes, you have a polar head for a phospholipid, and then you have two tails. Those tails could be essentially straight tails like this, or those tails could be bent or kinked like this. when the tails are straight, they are the result of fatty acids that are saturated with hydrogen. So every carbon has a full number of hydrogens. And so they tend to be very straight, and that allows the phospholipids to be packed really close together. So it allows this phospholipid layer to be denser, and there's less movement. And so at higher temperatures, the membrane tends to stay more intact. At lower temperatures, so this would be an advantage. I just got some new markers and they're all stuck together here. The advantage is for, say, cells at high temperatures. In contrast here, you would have kinked or bent fatty acid tails, which would be helpful at low temperatures. So advantage for low temperatures, and that's because the bent tails allow more space and movement and flexibility between the phospholipids of that bilayer that makes up the membrane, and so at really low temperatures where there's not much movement and everything would tend to be more dense, that there's still flexibility and fluidity to the cell membrane because of them. Right, so alterations to the phospholipids within the cell membrane you'll find at different temperatures. So organisms that survive and live and require low temperatures will tend to have more of the unsaturated, so these are unsaturated or polyunsaturated fatty acid tails. Those at high temperatures will tend to have the saturated fatty acid tails. So that's one advantage. Or one adaptation that you might find. Another is that, say, for the thermophiles, as far as proteins go, you'll find a lot more disulfide bridges. So there's a particular amino acid, the amino acid cysteine, which has a sulfhydryl functional group attached to the alpha carbon. And that sulfhydryl functional group can then form a covalent bond with another sulfhydryl group of another cysteine. And that covalent bond then greatly stabilizes the three-dimensional structure of the protein. So at higher temperatures, it's a lot more difficult to change the three-dimensional structure and have the protein break down. So you tend to find a lot more of those types of structures at these higher temperatures to keep the enzymes intact and functioning. And there's a whole variety of other types of biochemical adaptations, which is kind of beyond what I'm really trying to get into here. It's really just more the idea that different organisms have set ranges in which they live and can survive. There are some names simply associated with them. Psychrophiles for cold, mesophiles for these called middle temperatures, thermophiles for warm, and then hyperthermophiles for extreme temperatures. Usually we'll start to see a lot of the archaea, or other domain of life, at, really at all these ranges, but at some of the extremes. This is where we find a lot of them, and they have a number of additional adaptations. So for archaea, for example, their adaptation for, not all of them live at high temperatures, so don't mistakenly think that, but they have slightly different variation to the chemistry of their phospholipids, and what they'll also have is, especially for the ones at high temperature, bonds between the tails. So the tails are actually bound together. So instead of phospholipid bilayer being, where the tails are not, they're associated with one another because they're nonpolar and they don't like water, but they're not attached to each other. Here they actually covalently bound. So it really forms more of now it's technically called a monolayer because they are fused together. Although there's really polar heads at each side of it, it's one unit like that. So that's another adaptation that typically you see in archaea to the very high temperatures. We have some other types of environments like, say, there's freshwater environments, and we have ocean environments, right? So we have organisms that prefer different salt concentrations and, different pH concentrations. So you'll see, in terms of rates of growth, all different sorts of curves that you can find. Trying to break these apart here. It's a little more difficult than I thought. For different groups of organisms that prefer a variety of different types of salt concentrations, they'll each have sort of optimal salinities. So for a percent sodium chloride or parts per thousand measure sodium chloride. So those that prefer really high salt concentrations, we call these halophiles. So that's really a high salt concentration. Same thing we could find, I run out of room to keep drawing graphs here, but is in terms of pH. So pH preferences. So we have acidophiles, ranging from very low acidities. Say something like 0 to 5 pH. We have our neutrophiles, preferring kind of a lot more like neutral pHs, say like pH 5.5 to like pH 7, 7.5 like this, and then we have alkylophiles. Which prefer a lot more basic, conditions. So here we have things that are like, say 8.5 to like 11 in terms of pH. Running out of room to write there. So these prefer, these basic conditions. These prefer acidic conditions. And these are more neutral conditions. Now, what we're talking about here, though, keep in mind, this is conditions of the environment. Their internal, their cytoplasmic pH is roughly going to be neutral. So around something like, pH 7. For all of them, for acidophiles, for the alkylophiles, they're all going to have more neutral interior pHs, but it's just that they can survive in environments with very different hydrogen ion concentrations and or prefer those environments with those different hydrogen ion concentrations, and then their transport mechanisms will be different kind of based on that and other characteristics of their outer parts of the cell wall and structures that exist beyond the cell wall so they can exist in those environments. So overall, what you should be knowing, what you should know, or from this, is just some of the basic terms, kind of what they mean, the general kind of ranges, or rules for pHs and temperatures that the organisms prefer, and just some ideas of why they would have those particular types of preferences. So these organisms that might live in this cold environment, in the Antarctic, in the oceans there are a lot of different organisms that live underwater. People don't think there would be at say -2 degrees centigrade, but there are, and their metabolism is adapted to those cold conditions. People once thought that the organisms that were in the Antarctic, like sea stars and corals, would grow at much slower rates than their counterparts on tropical coral reefs. But we found that that's not true. They grow at pretty much the same rates, and their enzyme activities are pretty much the same rates of activities for the reactions, even though they're at very cold temperatures. And that's because they're biochemically adapted to those specific temperatures. And we see that in animals, but we see that even more so among the bacteria, because they can exist in environments where we just really don't find typically very, very many animals, especially at some of these great extremes. And so it's a lot of the different types of unique biochemical adaptations that they have to live in these different environments. So these are the factors that, part of the environmental factors that will affect their growth rates.